A drug went from concept to clinical candidate in 18 months. No test tube ever touched it. No cell saw it until it was already predicted to work. A computer looked at a protein, modeled its three-dimensional shape, and generated a molecule that nobody had ever synthesized before.
Wait, a computer invented a drug? An actual drug that goes into a person?
No cell cultures means no consumables budget. No consumables budget means I am paying very close attention right now.
The molecule didn't exist. The AI made it up, so it hallucinated a chemical structure, except instead of being wrong, it fit perfectly into the protein that drives the cancer. And now it's in a human trial.
The AI hallucinated a cancer drug?
It is mildly concerning that hallucinated and it worked are appearing in the same sentence and no one has paused.
How does a computer even know what a protein looks like? Proteins are invisible. They're microscopic.
We have experimental methods, X-ray crystallography, cryo-electron microscopy.
And now AlphaFold, which predicted the structure of 200 million proteins in a year, which means we can dock to proteins we have physically never seen.
Dock? What are we docking to what?
It's like parking.
So we're doing drug discovery for essentially free, computationally. What's the IP situation on a molecule the AI invented? Because if it's unpatentable...
The molecule came from a machine. Courts are still arguing about whether a machine can be an inventor. Meanwhile, the drug is in trials.
How many molecules can it even generate?
The estimated size of drug-like chemical space is 10 to the power of 60.
I don't know what that means, but it terrifies me.
It means we've synthesized maybe 10 to the 8 compounds in all of human history. We've explored something like 0.00001% of what's out there.
Stop! Nobody's generating anything yet. Before we get to the part where AI invents antibiotics, we need to establish one thing. How does a molecule bind to a protein? Why does that binding matter? And how does a computer predict whether a molecule it has never synthesized will actually work? By the end of this episode, you will understand exactly how a protein structure on a server becomes a drug candidate in a clinical trial, and why that process is simultaneously the greatest acceleration in medicine and a very compelling argument for extreme caution.
Welcome to the Krusty Lab. I'm Bobby Sponge, this is episode 11, and today we are doing in-silico drug discovery, computer-based drug design, molecular docking, and the generative AI that is inventing medicines nobody has ever imagined.
In-silico drug discovery, using computational methods, structure prediction, docking algorithms, and generative molecular models to identify and optimize drug candidates before a single compound is synthesized in a lab.
This is the most radical shift in drug development since recombinant DNA. We went from screen 10,000 compounds in 96 well plates and hope to generate 10 million novel molecules, rank by predicted binding and ADMET, synthesize the top 20. In months instead of years.
It is mildly notable that the field that took 15 years and $2.6 billion per drug is now partially outsourced to a GPU cluster.
Partially is doing a lot of work in that sentence, but I'm choosing to be optimistic.
Before we go further, if you're watching this and not subscribed, I have already modeled the binding pose of a molecule specifically designed to activate your regret receptors. Excellent oral bioavailability. Crosses the blood-brain barrier. Subscribe now, or I'll optimize the lead series.
Let's start with the target. A drug works by binding to a protein, specifically at a defined location, the active site, or an allosteric site, and by doing so, it changes what that protein does. It might block an enzyme from catalyzing a reaction. It might lock a receptor in an inactive conformation. It might prevent a viral protease from processing the proteins a virus needs to replicate.
So proteins are the bad guys.
In disease, often yes. A kinase that fires when it shouldn't, driving cancer cell proliferation. An ion channel stuck open, triggering epilepsy. A viral enzyme that the pathogen needs and the human host doesn't. The protein is the target. The drug is the molecule you designed to interfere with it.
And the beautiful thing is, proteins have shapes. Three-dimensional shapes with pockets, grooves, and clefts that are chemically unique. If you know the shape, you can design the molecule that fits.
Like a lock and key?
Emil Fischer described it that way in 1894. The modern model is more nuanced, induced fit, where the protein actually flexes as the ligand approaches. The binding is more like a handshake than a rigid key and lock.
The protein reaches out, the molecule adjusts, they settle into each other like they were made for it.
Made for each other. That's a brand story. I'm writing that down.
It is mildly romantic that the foundation of modern pharmacology is a Victorian metaphor about locks.
The binding pocket is where the action happens. It's a cavity or cleft in the protein's three-dimensional structure formed by the folding of the polypeptide chain. Inside, you have specific chemical features: hydrophobic patches, hydrogen bond donors and acceptors, charged residues, sometimes metal coordination sites.
So the pocket is specific. It's not just any hole.
Exactly. Imatinib, the drug sold as Gleevec, fits into the ATP binding site of BCR-ABL kinase because it mimics the geometry of ATP, but it fits better, well enough to outcompete ATP and block the kinase.
And that one kinase was driving the cancer. Block it. Stop the proliferation. Gleevec turned chronic myeloid leukemia from a near-certain death sentence into a manageable chronic condition because of one very precisely shaped small molecule.
One molecule, one pocket, it stopped a cancer.
I'm calculating the market cap of knowing exactly which pocket to fill. The number is large.
The point is, shape specificity is everything. The drug must complement the pocket's geometry and chemistry. The wrong shape, the wrong hydrogen bond acceptor, even a single methyl group in the wrong place, and you've lost the binding.
The pocket knows what it wants.
That's either profound or completely meaningless, and I cannot tell which.
So if you know the pocket, you can search for molecules that fit. Which brings us to how a computer actually does that search. Molecular docking. You have a protein structure, you have a binding pocket. You want to know, will this molecule bind? How tightly? And in what orientation? The docking algorithm places the candidate molecule inside the pocket and samples millions of possible positions, orientations, and conformations.
All on a computer.
All in silico. The protein is a fixed three-dimensional structure, a PDB file from the protein databank, or increasingly a predicted structure from AlphaFold. The molecule is represented as a three-dimensional structure with rotatable bonds. The algorithm systematically explores the configurational space inside the pocket.
Rotate it this way. Flip it. Flex this bond angle. Try every torsion. Millions of configurations per molecule.
And then what? How does it know which one is right?
The scoring function? For each configuration, each pose, the function estimates the binding free energy. How much does the system gain in stability with the molecule bound? The lower the energy, the tighter the predicted binding, the more potent the drug candidate.
Autodoc Vena. Free software. You can run it on your laptop. I have run it on machines in my shed that I am not legally permitted to describe in more detail.
It is mildly reassuring that molecular docking has reached the point where it runs on hardware of dubious legal status.
What exactly does the scoring function calculate?
Van der Waals interactions, electrostatics, hydrogen bonding geometry, the hydrophobic effect, the tendency of nonpolar groups to avoid water and cluster together in an aqueous environment, and in more sophisticated approaches, entropic penalties for restricting the molecule's conformational freedom upon binding.
Translation. It calculates how badly the molecule wants to be in the pocket versus floating free in solution.
That is crude but functionally accurate. Virtual screening is what happens when you apply docking to not one molecule but millions. The Eneman Real Library contains over 6 billion purchasable compounds. You dock all of them against your target, rank by predicted binding affinity. The top scores go to wet lab validation.
6 billion compounds?
Screened in days, with enough compute. Your wet lab then validates the top 50 to 100. You started with 6 billion. Your filter was a computer. Those 50 are the best of 6 billion. That's an absurd funnel.
How long would it take to do that physically, with actual test tubes?
High-throughput screening, HTS, the robotic 96-well plate system, can process a few million compounds at significant cost. 6 billion is physically and economically impossible by traditional methods.
So we just cut front-end discovery costs by, conservatively, 90%.
Conservatively, yes.
I need a moment.
Take your time.
No, I'm fine. I'm already structuring the deal in my head.
Okay, so this seems perfect. What's the catch?
The scoring functions are approximations, and the protein, in most docking runs, is treated as rigid.
Which it absolutely is not.
The rigid receptor approximation. Most docking software freezes the protein structure. The protein doesn't flex, doesn't adapt, but we just said binding involves induced fit.
So you're solving the wrong problem.
You're solving a simplified version of the right problem. Induced fit docking, where protein residues are allowed to move, exists, but it's computationally far more expensive. The rigid approximation makes the virtual screening of millions of compounds tractable.
There's also the water problem. The binding pocket in solution is not empty. It's packed with ordered water molecules. When the ligand binds, it displaces them. Displacing organized water releases entropy. Those molecules gain freedom, and that contributes favorably to binding energy. Calculating this accurately requires free energy perturbation methods that are orders of magnitude more expensive.
So the scoring function misses that?
It approximates it, empirically, which means the score is a prediction, not a measurement. In practice, roughly 10 to 40% of top-ranked virtual screening hits actually bind when you test them in a real biochemical assay.
So you dock 6 billion, pick 50, and maybe 5 to 20 actually work?
Compared to random screening, which gives you maybe one active per 10,000 compounds, that is still a revolution.
It is mildly characteristic of computational biology that the method works well enough to be transformative and not well enough to be trusted without validation.
You're looking for something you haven't found yet, but you sort of know which direction to look.
That is correct.
I had a drawer like that once.
And all of this assumes you have the protein structure to begin with, which for the past 50 years has been the hardest part of the whole problem.
We need to talk about AlphaFold.
What's AlphaFold?
In 2021, DeepMind published AlphaFold 2, a deep learning system that predicts the three-dimensional structure of a protein from its amino acid sequence alone, at accuracy comparable to experimental methods. They then released predicted structures for essentially every protein in the human proteome. 200 million proteins across all sequenced organisms. Free. Public database.
How long did it used to take to get a protein structure?
X-ray crystallography. Months to years, and only if the protein crystallized at all. Many medically relevant proteins cannot be crystallized. Membrane proteins, intrinsically disordered proteins, proteins that only exist stably when bound to partners.
The dark proteome. Thousands of drug targets for which we had no structural information. We literally could not see them.
And AlphaFold just gave us all of them?
In a year. A neural network trained on all experimentally solved structures predicted what decades of crystallography couldn't. That's not an incremental advance. That's a category change.
The entire structure-based drug discovery market just received a key to a room it couldn't enter. The firms that positioned around this in 2020.
It is mildly predictable that Crabzilla measured an epical achievement in structural biology by who captured the financial upside.
I also appreciated the science in parallel.
AlphaFold 3, published in 2024, extends prediction to protein ligand complexes. You can now predict directly how a small molecule docks to a protein without running a separate docking algorithm. The model outputs the complex structure.
And DiffDock applies a diffusion model to the problem. You give it the protein and the molecule, and it generates a probabilistic distribution of binding poses. Not a single predicted conformation, but an ensemble with confidence scores. It's treating docking as a generative problem rather than a search problem.
Wait, it doesn't just give you one answer, it gives you a whole range of possible answers?
With associated likelihoods, which is closer to physical reality, binding is not a single frozen state, it's an ensemble of conformations.
That's kind of beautiful, actually.
And terrifying. And we can do it in seconds on modern hardware.
Knowing how existing molecules bind is powerful. What happens when you stop asking which existing molecule fits and start asking, invent the molecule?
This is the part I've been barely containing myself about. Instead of docking a library of existing compounds, you design new molecules from scratch. Molecules that have never existed anywhere, specifically engineered to fit your binding pocket and pass your filters.
How does an AI design a molecule?
Start with representation. Molecules can be encoded as SMILE strings, simplified molecular input line entry system, a text notation that describes chemical structure linearly, or as molecular graphs where atoms are nodes and bonds are edges. Deep learning models can operate over both representations.
A variational autoencoder learns a continuous molecular latent space, a mathematical landscape where similar molecules cluster together. You sample from that space, decode the sample, and get a new molecule. Then you optimize, move through the latent space toward molecules with better binding affinity, better pharmacological properties, feasible synthesis routes.
What kind of properties?
ADMET. Absorption. Can it cross the gut epithelium and reach systemic circulation? Distribution. Does it go where it needs to go, and does it reach therapeutic concentrations at the target tissue? Metabolism. How fast does your liver cytochrome P450 enzyme system break it down before it reaches the target? Excretion. Can your kidneys handle clearance? Toxicity. Is it killing something it shouldn't?
So you can engineer the perfect molecule for the binding pocket, and it still gets destroyed by the liver.
Approximately 90% of clinical trial failures are attributable to ADMET problems that weren't caught early.
90%.
The phrase in the field is ADMET cliff. The molecule looks perfect in silico, then it goes into a biological system and falls off.
It is mildly apt that the final barrier between a computational drug and a real drug is a liver enzyme that has no interest in your docking score.
Which is why the best generative models optimize for binding and ADMET simultaneously. AstraZeneca's reInvent platform uses reinforcement learning. The agent generates SMILES strings and receives a reward signal based on predicted binding affinity, ADMET compliance, synthesizability, and novelty. The agent learns to navigate molecular space toward the corner where all four properties are simultaneously favorable.
Does that corner actually exist?
Sometimes, and when you find it, you have a clinical candidate in weeks instead of years.
Weeks versus 15 years. Just to reiterate, weeks.
In-silico medicine identified a CDK12 inhibitor for cancer. Full AI-driven program from target to clinical candidate in roughly 18 months. It's now in phase 2 trials. Hallicin, discovered by an MIT deep learning model trained to predict antibiotic activity, identified a compound with a completely novel mechanism against antibiotic-resistant bacteria, including C. difficile and Acinetobacter baumannii. A human medicinal chemist would never have prioritized it based on its structure. The model didn't know what an antibiotic should look like. It found what worked.
The AI found an antibiotic we didn't know existed.
In chemical space we already had access to. There are still billions of novel molecules we haven't generated at all.
It's like someone finishing your sentence, except the sentence is a molecule, and it cures something.
Finishing the molecular sentence, that's a pitch deck. I'm working on the deck right now.
Okay, but if we can generate any molecule, predict how it binds, and predict its ADMET properties, are we basically done? Is drug discovery a solved problem?
No.
Emphatically no.
It is mildly optimistic that is this a solved problem is a question this room is asking about drug discovery.
The scoring functions remain approximations. They still miss the full entropic landscape and the role of explicit water. The ADMET models are trained on existing drug-like compounds and may not generalize to truly novel scaffolds with unusual physicochemical properties. Synthesizability predictions can fail. A molecule the generative model scores as accessible may require reaction sequences that don't exist or don't reliably work. And none of it predicts what happens in a whole organism. In vivo pharmacokinetics, off-target receptor activity, immunogenicity, drug-drug interactions.
The model can predict binding. It cannot yet predict what happens when you give it to a rat, and then extrapolate from rat to human. Those steps still require biology in the real world.
Which is why clinical trials remain unavoidably expensive, even when discovery is fast.
In-silico has accelerated the front end dramatically. Target identification, hit discovery, lead optimization, faster by orders of magnitude. But clinical development is still biology. Still 3 phases of human trials. Still takes a decade from candidate to approved drug.
For now. 5 years ago, I would have said the same thing about protein structure prediction.
So we sped up the fastest part and the slow part is still slow?
Discovery used to take years. Now it takes months. Development takes a decade. That ratio is genuinely better, but But the hard part, proving it's safe and effective in human, remains irreducibly biological.
I'm building a model in my shed trained on failed Phase 2 trial data to predict clinical dropout risk. I will not say more than that.
It is mildly comforting that at least one person in this room is focused professionally on predicting failure.
Patar, we've been at this for a while. What do you think in silico drug discovery actually is?
You want to stop a protein from doing damage. The protein has a shape. The drug needs to fit that shape. You can't test every possible drug against every protein physically. There are too many of both. So you put the shapes in a computer and let it try all the combinations. Then you teach the computer to invent new shapes instead of just trying existing ones. The best shapes go to the lab. The ones that also survive the liver go to patients.
Patar, that was the entire episode in 6 sentences.
That is, I will not say it's more precise than what I said, but it is also not wrong.
The ones that also survived the liver. She really landed it.
The liver is undefeated.
The liver is undefeated. That's a t-shirt. That's a hat. That moves units at a biotech conference.
It is mildly humbling that the clearest summary of computational pharmacology came from someone who spent 40 minutes comparing molecular binding to parking.
Okay, final exam. We went extremely deep today, and I need to know if any of it stuck.
I'm inserting the mechanistic question, non-negotiable.
Pause the video right now. 3 questions. Answer them. Then press play for the discussion. Here are your questions. 1. In molecular docking, what does the scoring function calculate, and what does a lower score indicate? 2. AlphaFold 2 solved what problem that had been open in structural biology for roughly 50 years? 3. What does ADMET stand for, and which property represents the most common cause of clinical trial failure?
Take your time, no pressure, think it through.
We'll wait.
Question 1. The scoring function estimates the binding free energy of a ligand pose in the protein active site, accounting for van der Waals interactions, electrostatics, hydrogen bonding, and hydrophobic contacts. A lower, more negative, score indicates a more stable complex, meaning tighter predicted binding, meaning a more potent drug candidate.
I said it tells you how well the drug fits in the pocket. I'm calling that correct.
That's correct.
Finally, I got one.
I said it estimates the return per candidate, which maps perfectly onto DG as a proxy for potency, and potency as a proxy for commercial value. I'm taking the point.
It is mildly creative to express binding free energy as a return on investment metric.
Question 2. AlphaFold 2 solved the protein structure prediction problem. Given an amino acid sequence, predict the 3-dimensional folded structure. This had been considered one of the grand challenges of structural biology since Onfinson's work in the 1970s. AlphaFold 2 solved it at accuracy comparable to experimental methods, then released predictions for 200 million proteins publicly.
I said figuring out what proteins look like from just knowing their sequence. That is correct, and I am very proud.
It is correct.
2 for 3. This is unprecedented.
Question 3. ADMET. Absorption, distribution, metabolism, excretion, toxicity. The primary cause of clinical trial failure is metabolism, specifically hepatic first-pass metabolism via cytochrome P450 enzymes. A compound that is rapidly metabolized never reaches therapeutic concentrations at the target site, no matter how perfectly it binds in silico.
I said toxicity. I thought toxicity was the one that was the most dangerous.
Toxicity kills you. Metabolism just kills the drug before it can help you. Different failure mode.
I was so close. 2 out of 3.
I said excretion, because once you've sold a drug that clears fast, you've built in a compliance problem that guarantees repeat purchasing, which is pharmacokinetics as a business model. I maintain this is the most honest answer.
Mechanistically wrong. Commercially coherent.
It is